Hi, I'm Bob Schuster. I'm with DEP. I had a long career at marine water monitoring, have now moved into watershed and land management as watershed coordinator. I'm grateful to be here speaking about this today, as we've been involved with Barnegat Bay for a very long time, both the shellfish program side of my career and moving forward into, the nutrient side. Right now, what we have is monitoring of the tributaries and estuary. It continues. It was mentioned we're now in the third year of a third crop of a 5-year crop. This is to study all of the nutrients and water quality parameters that we started at the beginning. So one of the key points of why we're doing this and why we continue this monitoring, as all of those plans that you hear about are being implemented through 319 grants, how would we know if there's anything changing if we don't have all of that data and continue to collect that data? So the long-term monitoring as we issue out these grants is a very key component, because we want to know what changed, how much changed, and if we're seeing that benefit and what works and what doesn't work. All of the samples and collection of the marine samples are done in marine water. We have freshwater and biological monitoring DEP doing the tributaries, as well as a few partners that are still engaged. But having them all analyzed at the Leeds Point lab is keeping consistent with all the previous data that was done throughout that intensive part that developed the TMDL. We had 4 buoys originally. We added a 5th. We now have it in the Matitaconk. I will show a map shortly. But we also support 3 working with Barnegat Bay Partnership to host them on the same web page. So there are 8 continuous water quality monitors throughout the bay that will get us 15-minute data for pH, salinity, dissolved oxygen, temperature. So this is an ongoing effort. We're going to continue this effort because, again, we need to keep the information flow coming in to know if things are improving and how things are changing over time. The advantage of continuous is it gives us that diurnal change that you might not get in a grab sample. It gives us that seasonal, how it changes throughout a day, through season. So it's very important to have that to know what's going on. So all of this is, to monitor and see what's changing if things were implemented and what happens, and to identify hotspots. One of the things that we want to do is we want to look at those changes. We want to know throughout the estuary, are things getting worse, better? Are we seeing hotspots pop up that maybe we didn't see before? This is an important component that will be discussed probably a lot more in the 3rd session with implementation, because we want to make sure that wherever we're implementing things is in the areas that are the highest priority of where we're going to see the most benefit. So it's something that we're certainly looking to do. Throughout the estuary, these are the stations that continue to get sampled. We're sampling them monthly. So it's throughout the tributaries. I would like to mention that in addition to this, there's also a lot of ambient data at other locations that are not part of this monthly, initiative, that may be quarterly or other frequency, that is done by freshwater and biological on tributaries. That is also being done on marine water for shellfish classification. So there's a lot more information that we can also look at to look for problem areas in addition to what we're seeing here. And that's going to be one of the things that we're going to, look at, really doing that thorough deep dive, looking into that information. Continuous water quality monitors. There are a lot of them out there now. You see a lot of the orange dots or green dots are, if they're orange, they're not out anymore for the season. Green are still out. You can see the 3 green for Barnegat Bay Partnership are the land-based ones. The marine water ones are buoys. We tend to pull them so they don't get damaged in the wintertime. There's also freshwater lake ones, which were added for cyanobacteria blooms, which was something added a few years ago. They're all hosted on the same page. We're actually working with the New York Harbor Estuary Program, start adding more of continuous up into the harbor area besides just what we have in Raritan and Sandy Hook Bay. These are publicly available. So there's downloadable data that's archived. It can be pulled out, looked at by date ranges. Most of these in Barnegat Bay have been out since the 4 have been out since the beginning in 2011 of this initiative. One was added back, I believe, in 15 or 16 into the Matita. We're also going to institute a project that I talked about with some of the USGS staff here that we had conducted back in the late 90s up to about 1999. We did a land use study where we looked at 4 different land use types, and it was done under storm flow, dry weather conditions. Samples were collected throughout the hydrograph. And it helped us look at what the nutrient loads were from these different land uses and what was making it to the bay. This is something we're going to repeat. We have the QUAP is under review. It's being written. It's under review. Once it's finalized, we will start to collect those samples. It's something that is going to tell us whether, this fertilizer law that went into effect, have we seen a change? I mean, this was done pre-fertilizer law. This was done 25 years ago, some of it over 25 years ago. But once that law went into effect, are we seeing that change? Are we seeing nutrient levels change? What has changed? And how is it being impacted by that law? What we also want to do is I also want to reach out when I talk about hotspot identification. Something we're starting to look at, and we're starting to develop more tools for this, is we want to identify all available data, not just what we collect as part of this Barnegat Bay sampling, but there are a lot of partners. Brick Township MUA has routine monitoring. Freshwater has other locations. There's a lot of partners with data that all goes to the National Water Quality Portal. One of the things that we really want to look at is taking all of that data and looking at it over time, or looking at it over 5-year periods or 2-year periods. We can start to identify where some of these hotspots, where the highest levels of nitrogen are, where they are in the watershed. And that will help inform, as mentioned earlier, where where you might want to implement some of these plans, where you might want to apply for a grant, where we're going to see that hotspot location, where we'll see the most out of it. That's something we need to do. We're committed to continue to work. We want to work with local county governments. We want to work with the partnership. We want to keep that dialogue open, because when you go in and pull this data, believe me, you pull 30 years of data and you find out in Ocean County, just from nutrients alone, you probably have 200,000 water quality records. There's a lot to go through, and how to evaluate that, look at it for these hot spots. What can we do? How do we work together to identify where the spots are and take action? And we'll have a lot more to discuss in the future sessions on how to do that. But also keeping those partnerships, it's how we got here to develop the TMDL. Without all the partners collecting samples and partner labs at EPA and MATES, Brick Township, and all of the different partners we had engaged, we wouldn't have been able to get to where we are at this point. So I think keeping that dialogue open, keeping that, feel free to reach out. My contact information at the end, any questions at any time, people want to know more detail, I'm more than willing to work with people. We need to, because partnerships, no one group can cover everything. No township will have all the answers. The state won't have all the answers, but we work together, we're going to be a lot better off. So we need to keep that whole dialogue open. And it also keeps that transparency and sharing of information, so we all know what's going on, where it's going on, how it's going on. It's going to allow to make sure that we get to where we need to be. And by keeping everybody informed, keeping everybody engaged, we're going to be more likely to see success when we do implement these plans. I think it is a really important goal. I think Commissioner Vicari said it perfectly. Is in this room, and whether you're in the public or whether you're a municipality or county or federal, in the end, everybody wants to see the same thing. We want to see the water quality of Barnegat Bay improve, and the only way to do that is to keep that dialogue open and make sure we're all working together on the same page to get to where we need to be and want to be. And I think that's one of the most key points. This is my contact information. I will certainly share, answer any questions that anybody has.
So we did receive 2 questions. So we received a couple of questions. 2 were for Kim Seno from her presentation, but I'll do the questions for Bob first. So there was one from Maisie. What changes have you seen since implementing the fertilizer laws?
The fertilizer law was implemented, and we're going to conduct that study to see if we see those changes based on that historical data. So I don't think we've done a full evaluation to see of where that is, but by going to those specific land uses, when we redo that study, we're going to get a better handle on how that law has worked.
Okay. Thank you. And then from Paul Shore, if the nature of an estuary is an ecosystem that place for aquatic organisms, how will that productivity be measured?
I have to think about that one a little bit on how that would be measured on, the ecosystem side, because the target right now is to look at a nitrogen threshold, and our goal is to get to that nitrogen threshold. I think there will definitely need to be follow-up, as far as ecosystem-wise on what that impact is in the future if we achieve this goal. If we get these reductions, what would, we would need to duplicate some of those efforts to see how effective it was in improving that biological component.
Okay, thank you. And then for Kim, it says.
Can I add a comment? I would just add, I actually now feel good that I added a slide to my presentation. We do have an agreed upon suite of holistic ecosystem targets for the Bay. And so I can actually share that list with everybody in a few minutes.
Thank you. Okay, and then for Kim, let me just make sure. Okay. Are the MS4s tier 1 or tier 2, the NJDEP NJDEP sources? In other words, are the CWA regulated MS4 point sources?
They are treated as a point source. So it's a non-point source loading for the TMDL calculation, but for the MS4 permit, well, the tier A, everything's a tier A permit now in New Jersey, it is treated through that as a point source. It's getting permitted through the MS4 program. We'll explain that more in greater detail at a follow-up session.
Thank you. And Charles Caruso asks, can the TMDL encourage the adoption of a stormwater utility as an additional measure to help pay for the improvements needed to meet the nutrient standards?
I'd like to dialogue with folks on that at a future session, and we'll dialogue on that in the department. If that does appear in the TMDL implementation plan, it wouldn't be a requirement. We'd have to be clear that that's a suggestion. We wouldn't go so far as to make that a requirement. But if there's support for it, we would want to acknowledge that in the TMDL document.
Thank you.
And I'd also like to add to what Bob was talking about, the mid-1990s Toms River study. We also did a similar study in the Whippany River watershed up in North Jersey and Morris County area. And so the plan is to repeat these land use studies that were done, since we have this valuable information from the before time period, before there was the stormwater regulations, before we were doing many TMDLs, before we spent millions of dollars in 319 projects, and before we had a statewide fertilizer law. So they have the ability to repeat these studies from the same locations and compare the data, and hopefully to be able to see what reductions, where they're happening, and the nature of that, I think is going to be not just interesting, but hopefully profound contribution to our rules and regulations and what it takes to restore our waters. They don't happen tomorrow. It happens over a period of time. And to have the ability to at least look at that and see what the data is telling us, I think is just really, really interesting. And so I can't wait for those two monitoring initiatives to get kicked off. And then we, the department, will be able to share the results with the public. So I'm going to turn it over to Rob Carl. And Rob will talk about the existing Metedeconk watershed restoration and protection plan. And I'd also like to mention that for the Metedeconk, we, the department, took the information that Rob and his team had put together in that plan, and we wrote a separate protection plan, which we submitted to EPA. So a few years back, a lot of the states got together and they basically complained to EPA. And they said, you're making us do all these TMDLs and these restoration activities. What about for waters that are not impaired and we want to keep them that way? That takes money and effort as well. And you, EPA, aren't giving us credit for that. So EPA said, okay, we'll give you credit for that. But you have to formally write it up, submit it to us, and then they review and they accept that. So we did that for the Metedeconk, and New Jersey was, I believe, the first in all the states to produce a report to that effect, submit it to EPA, and to have acknowledgement of that. So we do have that on one of our web pages. It's a little bit buried, but we'll re-acknowledge that when we put forth the Barnegat Bay TMDL. It's Rob.